Now let us look at the characteristics of peptide MHC interaction. The MHC molecules show a broad specificity for peptide binding, in contrast to the fine specificity of antigen receptor. Each MHC has a single peptide binding cleft that binds one peptide at a time. But each MHC molecule can bind many different peptides. That is, the binding is degenerate. Keeping in account that there are 6 different molecules of MHC in a heterozygous individual and about 10 to 20 different molecules of MHC2, so the individual may be able to present enormous number of protein antigen. So MHC1 molecule can accommodate about a peptide of about 8 to 11 residues, and MHC2 accommodates a bigger peptide of about 10 to 30 amino acids. So there is always a complementary interaction between the MHC molecule and the peptide. The residues or amino acids of the peptide that binds to MHC are distinct from those which are recognized by T cell receptors. This interaction between the residues both in the peptide and the cleft of the MHC molecule are always non-covalent. So these peptides are acquired through biosynthesis and assembled inside the cell. But keep in mind the MHC molecule cannot differentiate between self and non-self peptide. The T cell receptor recognizes both antigenic peptide and MHC molecule. The peptide defines fine specificity of T cell recognition, while MHC accounts for restriction of T cells. MHC associated antigen presentation is involved in determining the nature of T cell responses and immunogenicity of the protein antigen. Let us discuss them one by one. So the nature of T cell response is determined by the type of MHC molecules being involved. If MHC1 molecule are involved, they will activate T cytotoxic cells, resulting in its effector function. If MHC2 molecules are involved, they result in activation of T helper cells and its effector function. So this response is also dependent upon the origin of the antigen, whether it is cytosolic or vesicular. The second effect of MHC-associated antigen presentation is on the immunogenicity of the protein antigen. As we know from our previous lectures that epitopes are processed, or the whole antigen is processed by the antigen presenting cells, making it more immunogenic and results in increasing their avidity, because proteases degrade the antigen or protein into multiple different types of peptides, but majority of the T cells can recognize and bind specifically to few of them. Those peptides are called immunodominant epitope or determinant. So thus they elect a high level of immune response as compared to the one which are not processed by antigen presenting cell and are not presented by MHC molecule. Most importantly, the expression of a particular MHC2 allele in an individual determines the ability of an individual to respond to a particular antigen. So that's why individuals differ in their susceptibility to same disease. See how an immunodominant peptide is displayed by the antigen presenting cell and MHC molecules. So the first step, the antigen is internalized by the antigen presenting cell, that can be either macrophages or neutrophils, which this antigen is then processed by antigen processing and which generates multiple peptides. So among these, the one which best binds to the available class of MHC molecule is called immunodominant epitope. So this epitope is selected and this T cell respond to only the immunodominant peptide epitope, resulting in enhanced immune response. As a part of second concept checkpoint, you have to find out what type of MHC molecule and T cells are involved in case of infections caused by the following pathogens. That is HIV, Vibrio cholera, Plasmodium, and Streptococcus aureus. Try to find out the answers and we will discuss them in our interactive session. The key points of today's lecture are: number one, the MHC complex is a cluster of genes that plays an important part in development of immune response and discrimination between self and non-self. The MHC class 1 and 2 are basically antigen presenting structure that binds to peptide antigen and displays them to appropriate T cells. Intracellular microbes, viruses, and tumors are expressed by MHC1 molecule and are then killed by cytotoxic T cell. Extracellular infections involved binding of MHC2 molecules with T helper cells and activation of both cell-mediated and humoral response. Antigen presentation via MHC molecules determines the nature of T cell response as well as immunogenicity of the protein antigen. I hope you have understand the basic concept. Allah Hafiz.